Jadi ya tolong dimanfaatkan dengan baik kesempatan ini 3 hari ini. Mudah-mudahan bisa mendapatkan hasil yang kita inginkan bersama. Kemudian yang kedua, tentu saja support kami tidak hanya terputus sampai di workshop ini. Jadi kita ingin punya relasi hubungan yang panjang, Pak, Bapak Ibu. Jadi ke depan setelah workshop ini apabila ada kendala dalam pemurnian protein, kemudian analisis protein, itu jangan ragu untuk kontak kami. Kami ada, mungkin Bapak Ibu kenal ada Mbak Iza sebagai FAS kami yang biasa membantu para peneliti di BRIN, jadi bisa kontak Mbak Iza atau juga kontak Mas Adi yang mungkin setiap minggu ada di sini. Jadi kalau ada kendala atau apa bisa kontak kami. Mungkin itu saja dari saya. Mudah-mudahan workshop ini bermanfaat untuk Bapak Ibu. Sekali lagi mohon dimanfaatkan waktunya untuk bisa belajar banyak dari saya. Mungkin itu. Terima kasih. Selamat pagi, Bapak Ibu.
Ya, terima kasih, Bapak Andri Wiguna. Next, I would like to ask Dr. Dede Heri Yulianto to give us an opening remark to Dr. Dede. Thank you.
Thank you, Ms. Mbak Uci. Ladies and gentlemen, I will deliver my speech in Indonesia. Yang saya hormati Deputi Bidang Infrastruktur Riset dan Inovasi Badan Riset dan Inovasi Nasional, Dr. Yohan Hidayat. Yang kami banggakan kan kolaborasi kita di workshop hari ini dari Cytiva Indonesia yang dalam ini diwakili oleh Mbak Widya Utami dan tim, ya. Dan juga dari Laborindo Sarana. Terima kasih sekali salam hormat kami Pak Andri Wiguna, dan juga to our honorable speaker Dr. Satya. Thank you very much for coming again to Indonesia and giving your speech here. And also yang kami banggakan peserta workshop protein purification technique using AKTA system for structural biology application. Assalamualaikum warahmatullahi wabarakatuh. Pertama-tama saya hadir di sini mewakili Pak Kapus, sekaligus mewakili manajer program Structural Biology. Ya. Jadi program Structural Biology itu manajernya ada Pak Ahmad, Dr. Ahmad Fatoni, dan beliau berhalangan hadir pada hari ini karena ada halangan hambatan di kesehatannya. Kita berdoa pada kesempatan pagi ini juga waktu yang baik ini semoga Bapak Dr. Ahmad Fatoni diberikan kesembuhan dan juga diberikan kemudahan untuk bisa bergabung dengan kita kembali. Bapak Ibu sekalian, pertama-tama sebelum kami menyampaikan sambutan, kami ingin mengucapkan terima kasih yang sebesar-besarnya kepada Deputi Bidang Infrastruktur Riset dan Inovasi Badan Riset dan Inovasi Nasional, Bapak Iman Hidayat beserta jajarannya. Juga kami ucapkan terima kasih sekali lagi kepada teman-teman dari Cytiva Indonesia, dari Laborindo Sarana yang telah bekerja sama dengan kita selama ini. Dan juga kami mengucapkan terima kasih kepada seluruh panitia, Mbak Sandy, ya, dan kawan-kawan Mbak Yeni, Mas Andi, dan kawan-kawan Mbak Uci atas terselenggaranya kegiatan pada pagi hari ini. Bapak Ibu sekalian, jadi sebagaimana yang kita ketahui bahwa kegiatan biologi struktur ini sudah berjalan hampir 1 tahun ya. Kita mulai, dan ini adalah tahun pertama kita sebagai benchmark juga untuk kegiatan platform kolaborasi. Ya. Dan sebagaimana kita ketahui bahwa kita berharap kegiatan biologi struktur ini bisa berjalan dengan baik, lancar, dan mendapatkan output serta outcome yang kita harapkan. Nah, kalau kita perhatikan dalam kegiatan struktur biologi ini ada beberapa faktor kunci ya. Paling tidak ada 4. Yang pertama, kita perlu mempersiapkan mengisolasi dan memproduksi sampel kita ya, dan hal ini misalkan protein. Ya, protein kita perlu kita isolasi, kita produksi, baik itu dalam bentuk wild type atau rekombinannya. Nah, tahapan kedua kita perlu melakukan purifikasi protein. Gitu. Yang kemudian ketiga kita melakukan data acquisition dengan alat cryo-EM, dalam hal ini teknik single particle analysis misalnya dengan kalau untuk protein, dan yang terakhir kita melakukan data processing atau pengolahan data sehingga mendapatkan 3D structure dengan menggunakan software misalkan CryoSpark dan lain-lain. Nah, Bapak Ibu sekalian, jadi manajemen biologi struktur bersama BRIN sudah melakukan beberapa training dan workshop ya kegiatannya. Memang kita bergeraknya dari hilir dulu ya, dari teknologi hilirnya, yaitu kita pernah melakukan training dan workshop kegiatan single particle analysis. Bagaimana melakukan data acquisition untuk protein untuk mendapatkan image ya, data imaging. Sehingga kemudian kita juga pernah melakukan workshop atau training terkait dengan data processing menggunakan CryoSpark. Nah, sekarang kita mundur sedikit ke belakang, itu melakukan workshop untuk protein purification menggunakan AKTA sistem ya, yang bagaimana nanti kita mendapatkan targetnya adalah mendapatkan protein yang murni. Jadi kalau mau mundur lagi ke belakang, kita masih punya satu step lagi nih, bagaimana menyiapkan protein rekombinan ya, misalkan nanti bisa saja nanti kita akan mengadakan juga workshop untuk bagaimana sih melakukan penyiapan protein rekombinan, khususnya misalkan seperti yang dulu pernah disampaikan juga, kita punya sistem tagging khusus ya, misalkan yang punya ya Cytiva dan sebagainya, itu bisa juga kita lakukan untuk workshopnya. Nah, Bapak Ibu sekalian, oleh karena itu pada kesempatan pagi hari ini tentu harapan kami adalah pertama, peserta workshop ya, terutama yang hadir secara luring ini dapat memaksimalkan fasilitas cryo-EM khususnya yang ada di BRIN, dan ada juga fasilitas yang kita sebut dengan protein purification unit ya. Insya Allah ini sudah bisa running saat ini. Dan juga kita berharap karena ternyata PI untuk kegiatan biologi struktur ini banyak juga proteinnya yang di bawah 100 kilodalton ya. Padahal untuk cryo-EM khususnya untuk SPA itu diharapkan protein di atas 100 kilodalton. Oleh karena itu mungkin kita berharap juga ke depan BRIN akan memfasilitasi kita untuk menyediakan sarana untuk kristalisasi gitu ya. Ke depannya kita akan mudah-mudahan bisa mengharapkan ada kegiatan kristalisasi. Karena kalau sudah bisa kristalisasi, nanti kita bisa melakukan analisis struktur biologi dengan menggunakan microED juga di lab cryo-EM. Atau mudah-mudahan ke depan BRIN juga bisa memiliki alat yang namanya alat kristalografi gitu ya, yang bisa menganalisis untuk protein-protein di bawah 100 kilodalton. Nah, yang kedua, kami berharap para peserta ya bisa bersungguh-sungguh ya hadir dan mempelajari workshop secara hands-on ya. Kebetulan alhamdulillah sudah ada Dr. Satya yang kita tahu bersama expert di dalam protein purification ini, sehingga nantinya para PI yang hadir bisa benar-benar mampu melakukan purifikasi protein secara mandiri dan bisa mendapatkan protein target yang diinginkan. Yang ketiga, ya kami menyadari bahwa tidak semua peserta yang daftar itu terpilih untuk memiliki kesempatan bisa hadir secara luring di sini dan melakukan hands-on. Nah, kita berharap juga mudah-mudahan ya di lain waktu kita bisa melaksanakan kegiatan workshop ini dengan tema yang sama. Ya. Kemudian juga kita berharap teman-teman yang hadir di sini bisa menjadi trainer lagi gitu ya. Anggap aja ini TOT ya gitu ya. Artinya nanti teman-teman itu bisa memberikan pelajaran lagi kepada teman-teman yang tidak hadir gitu ya, kalau belum mengikuti kesempatan. Nanti sambil sama-sama belajar melakukan purifikasi di lab gitu ya, dengan teman-teman yang tidak memiliki kesempatan hadir di sini. Nah, target ini ya Pak Kapus sampaikan ya, sebisa mungkin mendapatkan protein target yang murni, meskipun waktunya cuma 3 hari nih, Bu Widya ya Pak Andri. Tapi diharapkan selama 3 hari ini kita bisa mendapatkan protein yang murni gitu ya. Meskipun nanti dengan berbagai teknik ya, tadi ada teknik kromatografi, purification, optimization, termasuk satunya adalah analisis karakterisasi sampai dia murni. Misalkan dengan menggunakan SDS space atau pakai size exclusion kromatografi misalkan ya. Nah, pun kalau seandainya tidak waktu 3 hari ini tidak mencapai protein yang murni nih ya, nggak dapat single band atau tidak bisa analisis SIC-nya masih tumpang tindih peak-nya gitu ya. Ya kita harapkan nanti ada lanjutannya lah ya, lanjutannya supaya bisa benar-benar sampai dapat protein yang murni, yang benar-benar ready to go, ready untuk dimasukkan ke IOIM gitu ya. Itu harapan kami semuanya ya, Bapak Ibu semuanya. Akhir kata kami menyampaikan selamat sekali lagi kepada peserta yang hadir luring di sini dan juga Bapak Ibu yang online untuk bisa mengikuti workshop ini dengan sebaik-baiknya ya. Akhir kata itu yang bisa kami sampaikan, dan saya diminta tadi mewakili Bapak Deputi, Pak Dr. Iman Hidayat juga untuk bisa membuka kegiatan workshop hari ini. Akhirnya dengan mengucapkan Bismillahirrahmanirrahim, acara workshop on protein purification technique using AKTA system for structural biology application resmi dibuka. Terima kasih. Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you, Dr. Dede, for the remarks. Okay, ladies and gentlemen, now we would like to have a photo session. To all participants, please activate your camera and for the host to take the screenshot. Okay, ready? I will count from first slide. Okay. Please activate your camera. Ready? 1, 2, 3. Next slide. 1, 2, 3. Okay, next slide. 1, 2, 3. Okay, thank you. Ladies and gentlemen, now we are going to start the presentation and discussion session. This session will be led by our moderator, Ms. Vidya Utami. To Ms. Vidya, thank you.
Okay, thank you, Ms. Susi. Maybe this material will be presented by our FAS, Satya, ASEAN FAS. So we will bring two kind of material. First is in the preparation for protein purification, and the second thing is related with the AKTA, which is we will hands-on in the afternoon after lunch. So yeah, Satya, it's yours. Jika ada pertanyaan, nanti di tengah-tengah, kalau mau di stop, silahkan. Takutnya kalau di akhir keburu lupa. Ya, jadi nanti bebas mau jika ada pertanyaan dalam bahasa Indonesia maupun bahasa Inggris, mangga aja disampaikan. Terima kasih.
Good morning, everyone.
Morning.
How are you? Nice to see you again. So as we shared, the agenda will be first is discussing the sample prep in the first presentation. And the second presentation will be AKTA system, and try to see how you can utilize it for your purification, right? Followed by hands-on that starts like today. Yeah, hands-on start right today, and we have a sample with us that has a specific concentration, and it has a requirement of purity that I will take you through by utilizing the system, right? But before anything, that just want to throw the question to the floor, that you are only doing proteins or nucleic acids are also part of samples? Proteins only or nucleic acids as well? Proteins only?
Gimana Bapak dan Ibu mainannya protein
Proteins
dulu
or just
atau
nucleic
sudah
acids?
ada yang juga nucleic acid gitu? Iya Bu, ada juga. Boleh bantu cungen, cungen, angkat tangan gitu. Jadi mungkin Satya.
Okay, so proteins, how many? Yeah.
Protein.
Yeah.
Protein.
DNA and nucleic acid. DNA and nucleic acid. Okay. Okay, sure. So we have the sample preparation part of the purification is quite crucial. So that is why we are spending one session for getting to know what are the crucial steps to consider before you start purifying it, right? So I'm going to flash this slide. I mean, we're going to share this slide first for your reference. Yeah, okay. So you can refer to this slide as a workflow slide. So there are sample preparation steps as and then you move forward, and it ends up in the purified biomarker at the end. Can you see my, yeah, I think. The fullest. Okay, anyway, so we have the sample basically comes from blood, tissues, bacteria, yeast, biological, media like urine, barley, plasma, serum. So we have the protein that is basically expressed in the media, right? So anything that comes from the media, the cells, you need to process it. What it has is millions of other proteins called the housekeeping proteins. That is basically needs to be removed and purified, right? If it's nucleic acid, you have plasmid DNA preparation, RNA preparation, and DNA preparation. So you have three kinds. So plasmid DNA preparation has its own steps that includes DNA cleanup, PCR, cloning, and sequencing, right? For RNA preparation, you have RT-PCR, again DNA cleanup, and finally gene expression and sequencing, and that leads to the biomolecule of interest. Right? So the steps are quite very similar, but it is very specific to the DNA or RNA. So this cleanup step is done by kits that can remove your genomic DNA that can only separate the plasmid DNA. So this cleanup is quite important. That is why it is at the end of the step. And finally, DNA preparation, because genomic DNA, you have to go for cell-free cDNA or genomic DNA. Total nucleic acid and whole genome amplification has its own steps that can be done by PCR, DNA cleanup, genotyping, or cloning and sequencing. So these are sample prep that is quite crucial to finally get the biomarker of interest. So this is generally a workflow. So these slides will be shared, so you can actually refer this later. Right? And for protein, on the other hand, you have cell lysis, protein extraction. Cell lysis is basically breaking open the cell and then taking the content inside. That can be done by mild or harsh conditions. Mild condition are using detergent. Harsh condition are using sonication that basically disrupt the cell membrane. Or there are many things that consider harsh, including 8 molar urea is one of them. But generally, the lysis or protein extraction can be done. Enzyme regulation, basically adding enzymes to degrade the other impurities. Higher model protein depletion, protein enrichment, and immunoprecipitation. Desalting and buffer exchange and cleanup is to remove the cell debris or to remove certain salts that are used for cell lysis. Desalting very crucial because it removes the lysis conditions and brings to the buffer conditions. And finally, after you desalt, you need to concentrate it because your volume is quite crucial for loading to the resin, right? The resin that ÄKTA is controlling has a specific volume range. For example, in SEC, it's 2%. For ion exchange, it's going to be 30 to 40% of the volume of the resin is the volume of the sample. So your volume of the sample is quite crucial. So you need to concentrate, right? That is concentration. Concentration also helps remove unwanted proteins. in the first place, like using the filters that we have several filters range that can help you remove molecular weight cutoff. Anything above the cutoff is removed, and anything below it is concentrated. And you can actually reduce the sample volume and concentrate at the same time, and purify at the same time, right? So that is the concentration step. That can be done by various purposes. For 2D gel electrophoresis, liquid chromatography, or 1D electrophoresis. And finally, after you finish, you need to identify the target sample, clean up again, concentrate, because you're going to take a specific purity and of the homogeneous sample, concentrate again because it's post-sample processing. So after you identify the purity, you need to concentrate the protein again for your final analysis step, which is either mass spec or cryo-EM. When you do come to the final step, the purity has to be very high. So that is why we recommend another step of concentration. So you can also control the concentration of the sample you want to analyze, either by cryo-EM or mass spec. Mass spec requires a pure sample and also requires less sample. So you need to concentrate the protein so that you get a good peak or hit, as we call, in the mass spec chromatogram, right? Finally, your biomarker of interest is analyzed or characterized, right? So this is a workflow, a general workflow, but it has to be tailored. It has to be modified for your specific sample of interest. So your sample has its own workflow. This is just the method that you need to modify for your sample, right? So I suggest you have a protocol for every sample, and it is not equal to apply for all samples. One sample works by precipitation, one sample works by chromatography, one sample works by filtration, or another sample works combining all the techniques, right? So it is very specific because protein molecules and DNA are very different because you do mutations, you do different kinds, different cells. So the cleanup step also different. Okay. Any question on this? You can always ask questions. Yes. I have further slides to show you on the face, but overall, a cytosolic or cytoplasmic cytoproteins and cytoplasm is proteins that are able to be solubilized, which means your protein is, so they are, whenever you take a target protein, it either can be in the cytosol or it can be in the membrane, right? Cytosol has a lot of liquid aqueous environment, which means the pH is around 6 to 7. On the membrane, it is basically embedded in the lipid bilayer. So cytoplasm means the protein is soluble or water soluble. Membrane is water insoluble but lipid soluble. So membrane proteins are very hard to solubilize, but cytosolic proteins easy to solubilize. It's already soluble. All you need to do is choose the buffer, make it more stable, and doesn't precipitate. Number one. And for membrane proteins like GPCR, G protein coupled receptor proteins, all the other small proteins or peptides that are stuck to the membrane, is quite hard. It is very hard to get it purified individually. So you need to use detergents. You need to use solubilizers, stabilizers. That is depending on your protein charge and structure and time. So first of all, you need to know the stability of protein or peptide in the membrane. First is whether we need to identify if it's soluble insoluble. Soluble cytosol, insoluble. First, identify. How do you identify? Once you break the cell, you basically quantify or do an SDS page of the soluble fraction. Insoluble fraction is when you spin down the cell. When you spin down the sample broken cells. When you spin down, the pellet is insoluble. The supernatant is soluble. If you do SDS, and if you find your target protein in the pellet, then it's insoluble. If you find the target protein in the cytosol, in the supernatant, it is soluble. Right? If you find in the pellet, it's a bit difficult to make it solubilized. It means you need to work on detergent like ammonia or urea or quantity hydrochloride, all kinds of family to make it soluble. Yeah. So then comes different steps in insoluble to soluble, a lot of steps. Yeah. If it's soluble, then it's a bit easier. All you need to do is buffer exchange to your target buffer, either TPS or TRIS, or your depending on the buffer that you want to study. Yeah. Did it answer the question? Okay. So first is identify whether it's soluble or insoluble using SDS. Then if your target protein in insoluble, work on different detergent or stabilizer to get your protein soluble. So after you work on the pellet, it should come in the supernatant by adding detergents. Yeah. I have certain steps in the in my other slides for you to help, but it is not restricted to only the slides. It's a lot of other detergents you can also try. You can go by references of papers that it's already published, like what kind of detergents they use to solubilize this. If you need not go do this by yourself, there's a lot of materials that can help you, or recommended materials to help you solubilize your insoluble proteins. Generally, this book is very old, and it's a protein sample prep workbook. That's during the GE time. GE time is now Cytiva. It's now Cytiva. Previously, it was under GE, the life sciences. So they have this work handbook. It's online. I can share the link with you. But this handbook has some recommended buffers to solubilize your protein in case it's not soluble. And this is just a recommendation. There's a lot of other things that you can find online by published papers that works on soluble. Right? Okay. I'm going to the next slide in the interest of time. Okay. So, depending on the cell type, it differs. Bacteria has its own membrane housekeeping proteins. Mammalian cells has its own housekeeping proteins. Yeast has its own protein complexity. So you need to design accordingly. You can purify either by using a tag, a free-weight of a tag. That also involves the cleanup of the protein. But anyway, so this is sample prep in this step before you do chromatography. Yeah, so all the way to we have anti-protein enrichment, and protein enrichment is basically pull the protein and prepare and concentrate the protein. So we have different resins, we have different consumables for basically 1D electrophoresis we also use magnetic for pulling the samples down and using it accordingly for the specific sample prep. So we have a selection guide. You see, for example, in terms of small scale that can help you concentrate your protein and actually just isolate your antibodies as a first step. So we have both centrifugal columns, and also we can actually have actual resin later in chromatography step. Desalting columns, like PD10 columns, can help you separate or remove salt without any by manual techniques. You can always choose it from our list of manual or catalog that can help you choose the target resin. choose the target consumable before you prepare your samples and be ready for the next step. Okay, so I'm going to share these slides maybe for your reference later, so you can choose and go through the specific kit for you to use for your sample. Okay, this is quite short, so it has its own DNA, RNA, and protein preparation and concentration step. You have triple preparation kits that has culture cells with starting quantity up to 5 million, 550 million, and animal tissues that can be done from 1 to 20 mg tissues. Downstream applications of cloning, gene expression, genotyping, and protein analysis. You have preparation kits that can help you do this, right? And for DNA cleanup, we have the basic PCR kit, hotspot PCR, and you have other genotyping, cloning, and sequencing and gene expression that has the corresponding beads that can help you do this. So we have PCR beads, we have ready to go rapid analysis beads, ready to go RT-PCR beads that can help you clean up your sample. All these are kits that comes with the reagents and the consumables that you want to prepare. Yeah, so in the meantime, so let me go detail in my other slides that can tell you the specific steps on the solubility. Yeah, this is, oh, sorry, there is a lag in the sharing of screen. Trying to see. Yeah, so this is introduction to protein sample preparation in specific, not just plasmid. We'll do proteins because majority of us do proteins here. And I will take this opportunity to take an example of protein sample prep for you to choose your workflow, right? So what we'll be checking is the overall overview and then strategy formats and challenges when it comes to sample prep. And we summarize to actually have it closed. Okay, sample prep workflow generally starts with the collection and extraction of the cells and enrichment, and followed by the protein analysis, right? All these 3 steps requires cleanup or conditioning, and that is specific for samples from the start materials to the homogeneous or to solution form, and actually finally prepared analysis sample that is ready for your mass spec, your cryo-EM, and everything, right? So this is a workflow that you need to optimize and customize and finally have it in your protocol. So collection and extraction specifically involves using techniques to break up the cells and the tissue architecture. We call it clarification, clearing basically, and cell disruption that involves grinding, even physical grinding in a mortar and pestle in a test tube. It's always possible to break it up and get your proteins out in the tubes, right? Clarification involves filtration, centrifugation. Filtration can be 0.22 micron filters to remove big precipitates and chunk. And then centrifugation that can involve our small, Vidya has some filters to show you that has cutoff membranes to remove the 3 kDa or 10 kDa proteins based on molecular weight, and that can help you clean up and remove your contaminants, and actually you can actually clarify at the same time. You can solubilize protein, you can stabilize them by using detergents, stabilizers. Stabilizers, very common example is glycerol. When you add it, it prevents precipitation of proteins coming together, right? And other things such as EDTA that chelates ionic free ions and removes it from the solution and prevent it from destabilizing. And we have protease inhibitors that basically prevents the proteases from chopping off your proteins. The cells has natural proteases that basically makes it unstable for your target protein. So you need to inhibit the proteases. If you have phosphatases, that can also be detrimental to your protein sample. You can have phosphatase inhibitors and reducing agents, chelators. All these are possible to finally prepare your sample. We call it the small scale prep because we are doing a small scale preparation, right? So top 3 techniques used in small scale is homogenization, solubilization, and stabilization. Homogenization is basically clarifying, breaking the cells. We call it homogenization step, or the equipment which is doing this is called the homogenizer, right? We have the detergent or lysis methods. We have gentle, medium, and very harsh condition. Very harsh condition is when it comes to sample which can never have soluble protein. You need to do a lot of harsh condition to extract the protein from the membrane. That's why it's called a rigorous or a heavy or tough sample. Tough sample is very hard to extract proteins out, right? Tissues are tough samples generally when you need to grind the sample very harsh and in your buffer and extract the protein. So very simple techniques, which is gentle, is many cells that you add lysis buffer to get your proteins, lysozyme for E. coli, and zymolase for yeast. Grinding is a moderate technique where you grind your tissues or samples in a mortar and pestle in the buffer of interest to get it done. Ultrasonication is the most toughest because bacteria and tough samples are very hard to break. Ultrasonication involves sound waves and also creates mild heat to basically clarify your samples. Right? Depending on how harsh your solubility is required, you can use the technique. Always refer literature. Step 1, if you have the same protein that is done already, or you can always check, handbooks like us. All the material that is already existing. And then if you don't have anything, you start by my medium. How do you know if your protein is existing to SDS? Of course, mass spec can be done, but SDS is easier. Right? Solubilization, kinase, detergents, chaotrops, and detergents is ionic and non-ionic. Detergents, which is non-ionic, is for intracellular proteins and non-denatured. Ionic makes it denatured. So, ionic is most common example is SDS, and that is a universal solubilizer. If it works in SDS, it's very simple for you, it's easy for you. But something which is very harsh is detergent. And Chaotrops.
Biasanya kalau kita bukan pasti
Chaotrops is basically exposes your hydrophobic patches and breaks it down. And detergents, just 8 molar urea, very harsh, can also be used to solubilize. So all these are different conditions where you can solubilize your protein, right? And stabilization. After you solubilize, you need to keep your protein stable for X number of days for your sample to analyze. Or you want to store it in the fridge, or you want to find out how you're going to use the sample after you purify, right? That's called the stabilization phase. Stabilization phase involves enzyme inhibitors, phosphatase inhibitors. You keep it active for a long time. Buffer control, avoid extreme pH is going up and down, and redox agents like DDT that prevents the sample against oxidation. DDT is commonly available chemical that you prepare in stock and add to your buffer at 1 millimolar final concentration to prevent oxidation of protein. Right? So, 3 technique is homogenization, solubilization, and stabilization. So, there's another step called There's another step called enrichment. Enrichment is for increasing detectability and also for reducing complexity. Increasing detectability is basically improving purity before your actual chrome step that goes for high scale purification. So increasing detectability is done by affinity capture, for example, of protein A or G kits that targets antibodies, nickel IMAC resin that targets histidine tag proteins, and TiO2 targeted phosphorylated peptides, and streptavidin that recognizes your bionegative tags on your protein. Pre-activated affinity resins can also be done. You can also activate the resin or create your own resins with specific tags, affinity for your tag proteins. It's called do-it-yourself. Depletion resins is also available, including albumin and immunoglobulin removal. Right? That one is specifically removing albumin. That's removing certain things are also called depletion resins. Reduced complexity is basically based on solubility. You do differential fractionation, like your sample of different natures inside, like soluble and insoluble. You can actually do differential fractionation. And finally, of course, you can use chromatographic techniques to reduce complexity for those which is hydrophobic and non-hydrophobic, small and big. That's basically reduced complexity. That can also use ultrafiltration, 1D phage, free flow, isoelectric focusing. All these stuff can reduce the complexity and get your samples to purity. Enrichment of antibodies, as an example, as protein A and G has different selectivity and subclass. And you can use these two kits basically to separate these two antibodies based on heavy chain and light chain. Fusion protein as well, histidine tags. In this case, you can actually see the use magnetic beads for minimal sample usage and for screening many construct with little sample. How do you tune it? By adjust imidazole content. With higher imidazole gives you higher purity, but at the same time, when you shoot the imidazole very high, your protein becomes unstable. So you need to choose a concentration that is not very toxic to the protein. At the same time, it's okay and keep it stable. Buffer exchange may be needed since imidazole is very strong. So you need to do it immediately after you purify or prepare your sample. Finally, is cleanup and conditioning. Ensure compatibility in workflow. Removal of low molecular weight substances. You exchange a buffer, you use desalting, you do dialysis, you do ultrafiltration, ultra diafiltration, and another one is another thing. So you can also do concentration of proteins using this filtration. There's a lot of kinds. You can do dialysis, you can do chromatography if you have that's the only choice. You can do precipitation. We have an example of ammonium sulfate precipitation that she gave us. We can add ammonium sulfate, precipitate it, and then remove it so that you can actually concentrate it as well. So cleanup and conditioning is, you have a lot of choices. The easiest is the desalting column. Just inject the sample into resin and take it out. Or dialysis, that you keep it in a beaker. And dialysis membranes comes with the cutoff 3 kDa, 10 kDa, or more than that, 100 kDa. You can use these membranes. You can keep it overnight in lab and clean up your samples to remove salts and other conditions. Bulk impurities such as sodium chloride, if you want to, bulk salt, if you want to remove after a specific ion exchange step, you can always use PD10 columns. You can do desalting columns, and this is a typical example where they remove NaCl from the albumin that is separated from 0.5 to 0.2 to 0.5 ml sample, 0.75 to 10 ml sample, 1.75 to 25 ml sample. We have the separation profiles that is basically showed here. Always try to keep protein losses low, because more steps, more loss, right? So try to reduce the steps as much as possible and minimize the number of steps in the workflow. Very standard rule to keep the number of steps low so that you can keep the loss low. Strategy, point 2. Purpose of protein sample prep is for enrichment, for analysis, increasing the detectivity of target protein. Transfer target protein and collection and extraction, and finally cleanup and conditioning to make sure that your protein is compatible. Planning is the key. So you need to first plan, keep the overall workflow minimal, choose the right analytical strategy, the amount and purity needed and state of the proteins, learn the limitation and capabilities of the analytical technique, know the sample source properly, whether the sample source is from mammalian, tissue, other samples such as blood, urine, plasma, all these has its own preparation step. Know your sample and choose the best format, of course. Best format is choose what tools to use, what kits to use. Even if it involves resin like cleanup or filters, please make sure that you do the right step. And even if it involves one more step of adding an ultrafiltration, filters to clean up your precipitates, please do. But make sure that you have the right strategy and customized protocol with minimal loss and minimal effort. Okay, that's involved in planning. Planning should not overdo it. Add too many steps is also overdoing. But in this case, keep the number of steps done. Work quickly through the steps. Always make sure that your protein is stored pure and it should not be stored impure, right? Once you break the cells open, you need to work towards purity very fast and store it. Storing is basically you can add stabilizer and keep it in fridge. It can be stored in 4 degrees or -20 or even -150 if the protein is really unstable, right? But you need to work towards storage and make sure that's stored at the right time. Okay, choose the analytical strategy, right? Analytical strategy, right? If you need to do mass spec, there are a lot of strategies that you might finally end up. You might do mass spec, you might do immunoassay, HPLC to make sure that you detect it in the right region or affinity, or functional study or structural study, right? Functional structural studies. So depending on which one you want to use, you need to know what is the technique. Can the technique be done with the presence of salt, or if it interferes with a lot of if the technique has a salt interference that it cannot be used, it doesn't allow. What is the concentration that is used for the technique? Okay, for mass spec, the concentration may be in micrograms, 0.1. I think. How many of you do mass spec in the room? Mass spec. Mass spec. Your for protein analysis. No one does. How about cryo-EM? Cryo-EM. Sorry? For analysis of your protein. Yeah, cryo-EM. So the point I'm saying is you need to know the concentration of protein that is detectable in cryo-EM. What is the range that you can detect? Can you do micrograms? Can you do milligrams? 1 mg per ml is the mass spec determination range. So that is, you need to know that concentration should be the final concentration before you do the sample prep. So after every loss, you will lose 10 to 20% of sample. That is very general rule. So if you want to end up in 1 mg per ml for mass spec, you start with something like 2 mg per ml in the beginning. Make sure that once you do the solubilization, your target protein is at least 2 mg or around that, right? So also need to know if your salt is affecting your mass spec data. Cryo-EM, for example, what are the interference? Does EDTA interfere in your detection? You need to know all kinds of background, particulates that can interfere with your assay. Very common example I can give is protein assays. You have 2 assays, right? Protein quantification is, you can either do by COMA-C, you can go by BCA assay, right? BCA assay. So, there are many interference in many compounds that can interfere in your BCA assay. Anyone knows the salts and anything that should not be used in BCA assay? BCA can be used. The BCA assay is bicarbonic acid assay that works by mixing your protein sample in the buffer, and then that data detects at 562 nanometer by spectrophotometer. But anyway, so my idea is to say that after you get the kit, you need to make sure that the reagent in the kit should not have interference in the buffer that you have, with the buffer that you have. Buffer, not the protein. So for example, there was one sample I was working before. The buffer has EDTA, but EDTA interferes with the reagent. EDTA can bind to the reagent and turning color. So you need to know it is not your protein, it's EDTA. So iron can also interfere. So every reagent has interfering salts. Please check that list of interfering salt before you design your assay. In other words, just put your buffer and see if it turns color. If it turns color, then it's not a good reagent for your assay. Change it. Please change it. Or use the right concentration. So you can dilute the sample or dilute the buffer with water, then you can actually do the assay reconstitution. So you can always find a workaround way to make sure that the interference by a background is not too much, right? Certain salts can turn up in mass spec, giving you a peak because of it binds to the resin and everything. So you need to know if your sample has a clean baseline. When I say baseline, your background should not have any reading. What is your background? Your background is a buffer. Your background is a stabilizer. Your background is your inhibitors that should not give you any signal. So your actual sample will give you signal, right? So this is quite important, but quite crucial. That is why choose the right analytical strategy to make sure you detect only the protein, not the contaminant. Okay. Formats. Oops. Yeah, formats for enrichment and cleanup and conditioning. We have different kinds. I mean, to help you do this, we have microcentrifuge, we have normal centrifuge columns, syringe filters, or or systems that can handle like small resins that can use. We have gravity trap columns with fixed amount of beads, fixed maximum sample volume, and magnetic device. We call them max up rows, and variable amount of beads and variable sample volume. And it's always very flexible and then automated. So enrichment and cleanup is something that has to be done with minimal effort, and we have the kits for you to choose to help you do this and clean up the condition. You can always check our brochures and protocols. Ask Vidya any gaps for products. She'll be happy to answer them and share with you the right brochure for you to choose. Manual handling of in my slide. Yeah, manual handling of various volumes, sample volumes are quite crucial. That can be 0.3, can be like 3 ml. So we should design the right or choose the right filter. We have both the small and big filters and we have just the small scale, as we call, is something that is fixed in the tubes, either in small 1 ml or 15 ml or 50 ml. We have the right volumes, and please choose the right selection. And it's the corresponding kits and filters that can help you purify your sample, right? Manual handling of very small volumes can lead to loss. So please be prepared and prepare excess proteins to accommodate for the sample loss that's going to happen in your purification process. This is an example, purification of monoclonal antibody from CHO cell activation media. Equilibrate, load sample, wash, and elute. Typical example is protein A, protein G, gravity trap. It can also be done by automated using vacuum manifold, where you add samples to the resin and use vacuum to take out the samples and elute the samples later. All these are protocols that is available online. You can always not just part of Cytiva, it can be anything that can be used with the use of vacuum manifold. This process of sample prep can be automated as well. Challenges when it comes to sample cleanup and sample use is at the sample harvest. You start with this stage where your sample is very active, stable, and after a few minutes, your ATP, glucose, and pH starts to act on it, and it goes ATP and glucose and pH level goes down, lactate and calcium level goes up, and it has an effect on the amount of sample, right? Calcium activated proteases in several minutes, it has an effect on your sample and you lose some more sample. And finally, your cells start to die because things are like working on it, and only your part of your sample is active over time. So changes in the sample nature as and then time passes, it's quite very crucial. So you need to design something that is based on your stability of your sample. So things can act and make it degrade and make it worse. So challenges in again post sampling is SNAP is basically loss of protein choice. So basically SNAP freeze or precipitate the sample to stop degradation if you think the sample is dying so fast. So if you can't process the sample immediately like today, you want to actually freeze it. Just snap freeze it, flash freeze, check on step as to how to store your sample, freeze it to basically get it done to prevent a stop degradation of sample going into loss. Right? Add protective compounds as soon as possible, inhibitors, stabilizers, proteases, phosphatases, and protect against oxidation. The major is DDT, prevent oxidation in your by adding it in buffer. Right? Minimize damage, avoid lengthy steps and also avoid lengthy times that the sample is kept in the lab at room temperature. Try to minimize your time when it comes to sample purification. Make sure that you freeze it as soon as possible. Detectability of low abundant proteins is another challenge. Enrichment can be done by using cyan dye, mix it with our dye reagents. Make sure that you can actually amplify this by using fluorescence tags, and that you can detect in 2D gel electrophoresis. Tags are the key that you can actually use it for detection. Right? Without enrichment and after enrichment, you are basically able to find the green spots that is your target protein by using specific site or tags that can bind to your protein. Enrichment for phosphopeptides, if that is your kind of samples that you end up with, is workflow analysis. For example, from leukemia cell lysis, trypsin digestion, desalting of peptides, using MAX approach, TiO2, like phosphorylated peptides protocol, and that can be helped to prepare your samples, and finally analyze of samples. We have specific enrichment protocol for specific peptides such as phosphopeptides, histidine tag peptides that you can choose for sample prep. Enrichment is, there's a lot of things that you can actually find. So the titanium dioxide binds to both the serine and the tyrosine and tryptophan phosphopeptides. You need to make sure that you identify the right compounds, the right affinity that you want to choose for separating and clean up your samples. Reduced metric effect from serum samples is you need to make sure that your task with quantifying specific binding activity of NAD in serum samples using SPR. So sample characteristics in this case is ultra high complexity because serum got a lot of complex protein. It contains many potential interfering compounds, and on top of it, it is very viscous. It is not very liquidy. It is very highly viscous. It's sticky. So binding levels need to be worked in terms of non-processed samples. In this case, you see the blue lines are turn up in your chromatogram or sensogram. The blue lines are basically non-specific binding. So this is because it has a lot of other complex proteins that show up in your data and that can affect your data as well. How do you clean up? Is by using enrichment techniques, cleanup techniques, immunoglobulin enrichment on magnetic beads and desalting in 96 well filter plate. And thereby you can actually see the binding level of processed samples with minimal blue background, which is a non-specific binding that is reduced when you do sample prep. Right? and you can also use protein Gmax of rows extra, where you can actually see the binding result compared to the reference is way more higher. Finally, summary. Right? Summary of what we've seen. Get quality results with minimal sample prep. Know your sample. Make sure that your sample nature is very clear. Check on literature to check the stability of your protein. How long does it last in what buffer? Any interference in the stability? Is it easily degradable by phosphatases? Make sure that you've got the right inhibitor, right? And you choose the right conditions for your sample. Learn the limitations. If the protein degrades over time, make sure that you know how long does it last before it finally degrades. Shelf life, as we call the protein for the protein sample, shelf life is the amount of time that it can actually be stable in a solution in a specific temperature, right? And finally, read the handbooks. We have a lot of handbooks that we have already. It's online. Ask us if you require any specific in-depth handbook. Handbooks for chromatography, handbooks for. We have handbooks for each and every step, like IEX, SEC. We have HIC handbooks and everything. So read the handbooks for detailed description of the components and resins, their nature and know-how. So for you to design. You can also use Purify app that is online to choose the right product. For specific ACTA, for specific, you can choose the resin for ACTA. So you have 3 ACTAs here, ACTA Pure, you have ACTA Pure N. So you can always use the Purify app to customize your resin according to the ACTA that you want to use. Okay. So I encourage this to use because it's a simple link. You don't have to install an app. It works directly on your web browser, and finally that can you use customize your products before ordering. Okay. Of course, we have purification handbooks from Cytiva. We have the links for the handbook. Vidya will share. Yeah, Vidya will share with you. You can always go through that. If you have any questions on specific topics, you can always email us, and happy to help on clarifying questions. Thank you. And this brings me to the end of the sample prep presentation. Thank you. And more questions from the audience? Any questions from the audience? Yeah.
Ya, mungkin Bapak dan Ibu, baik yang online ataupun yang offline di sini, gitu, cukup jelas materinya dan mungkin ada yang mau share perihal challenge yang sedang dihadapi gitu. Siapa tahu Bapak dan Ibu di sini sedang dalam proses yang sama, dalam proses preparasi untuk sampel ini. Feel free to share, atau jika ada pertanyaan juga silahkan bisa ditanyakan Pak dan Pak Bu. Bagaimana? Oh, sepertinya baik-baik saja kelihatannya. Mungkin kalau ini di chat box juga saya tambahkan link untuk handbook yang terkait dengan preparasi sampel. Di situ juga ada website dari Cytiva, di mana itu tersedia banyak sekali jenis handbook di situ. Apalagi perihal protein purifikasi dan teknik-tekniknya. Jadi nanti kalau ada waktu silakan bisa dibaca juga. Dan itu free access, jadi nggak perlu login atau segala macam, bisa langsung di-download. Oke, itu poin yang pertama, materi yang saya bawakan, betapa pentingnya proses preparasi ini gitu sebelum kita masuk ke tahapan purifikasi gitu. Nah, mungkin jika tidak ada pertanyaan, mungkin kita bisa lanjutkan karena tahap preparasi ini yang nanti salah satunya yang akan kita bahas atau kita lakukan hands-on-nya gitu di jam siang nanti gitu. Jadi mungkin semoga nanti ada ketemu pertanyaan-pertanyaan ya pas hands-on gitu. Jadi lebih kebayang dan lebih realistis lagi.
Okay. I continue to the next ACTA. Vidya.
Yeah, yes.
No. It's on. Oh, okay. Yeah, okay. Do you know what is the ACTA system that you have? ACTA? ACTA Pure 25? And? ACTA Go? ACTA Pure Micro, right? Good job, good job, good job. So, you know the ACTA system, right? But basically understand the capability very nicely. So you need to know when to use the right ACTA system. You have three ACTA systems, like Pure, Go, and M, right? Each of them has its own applications, its capability that you should not use the wrong ACTA system. I mean, you might end up in wasting buffer because you cannot do a large sample volume in ACTA Pure Micro. And you cannot do low sample volume in Go or Pure, right? You need to position the right ACTA system. So this presentation will be for positioning your sample to the right ACTA, so you can actually use it better and efficient. Okay, so sorry, this is not. So now you have both Pure and Go, right? So it supports your ACTA Go is for routine purification. So it has got one pump and it has got one sample inlet and a fraction collector. ACTA Go is when you want to have a buffer exchange step. If it can do one ion exchange step or his stack purification, 5 ml columns, or the highest you can SEC column, you can go 25 ml. So very simple one step can handle by ACTA Go, and the sample volume can be from 5 ml all the way to 50 ml, no problem. So choose the right ACTA. The 25 ml per minute is the ACTA Go pump capacity. So you can do flow rates as low as 1 ml per minute or even lower as 0.5 or 0.4 also possible. But it's for routine purification. No, you cannot optimize in an ACTA Go system. You know the protocol, you know the flow rate, you know the sample volume, ACTA Go can do no problem. But more when it comes to Pure is for optimizing conditions. Optimizing condition is like you have more buffer inlets. You can actually screen buffer. You can choose buffer, which you can actually work on optimizing buffer conditions as to which buffer elutes better. And you can also use ACTA Pure for injecting different samples at one time with sample pump. Because you have two pumps, A and B, you can optimize. You can choose different elution buffers, A as B as well. Choose different buffer conditions. What is the best elution buffer? What is the less binding buffer that you can optimize with A and B? So process optimization can be done way more easier and better with ACTA Pure. But same pump 25M, you have detector there, and you have, I think, U9M, right, Vidya? So you have multi-detector, multi-wavelength detectors. If you are using with tagged samples of fluorescence, you can actually detect the protein and the fluorescence, or even DNA with the ratio of 260 to 280 detectors. You know that you can actually 260 to 80 ratio is basically DNA content, and you can optimize the separation of genomic DNA from proteins, proteins from DNA from other protein contaminants. Target you can process optimize using ACTA Pure. Yeah, we have with different wavelength detectors. ACTA Pure L is single wavelength, T is 2 wavelengths, and M is 3 wavelengths. So you have M which is the multi-wavelength. That's the best condition ever. Please utilize it for separating tag proteins, separating DNAs. Utilize the multi-wavelength capability as much as possible. Okay, so you can optimize your conjugation, binding, and everything, separating from your ACTA Pure Micro is let me see. Yeah, this is how a pure looks like. Of course, you know, it gives you more chance to control automated workflow. You can automate multiple sample injections one by one. Method Q is you create method and you can repeat the method again without you being there in the lab. Queue the method and collect the samples. Maximum runtime, automated runtime without you being there. You can create all these methods and be very smart. You don't have to be there. You can do other things as well. Okay. ACTA Avant is our next step. How many of you know about Avant? I'll do a quick run through. Then we can go back to it. How many of you know Avant? Not much. So no, Avant is the next step of Pure. Avant is for process development. Process development is more optimization step in terms of productivity, yield, quality of protein, and with the software that helps you, called the design of experiment that comes with the software. Design of experiment can improve your process optimization parameters with reduced time that you spend in screening condition. You can screen pH, buffers, columns, screening columns. You can do a lot with the additional software called design of experiment that can give you good data at the end of the experiment that gives you heat map of high protein yield with concentrations with conditions that you try. To reduce your time optimizing parameters in the lab, ACTA Avant is the goal. It has automatic buffer preparation feature that can help you create buffers from the stock solution that you load and create different variants of buffers and screening conditions as well. Okay, so quick check on, you need to know what column to use on which ACTA. So basically, you should know the pressure difference that is to be tolerated on the bed whenever you purchase a column. Make sure that your delta, your pressure on the packed bed is of certain limits, and make sure that the pre-column pressure should not exceed the limit of the conditions of the resin. Please check that in your instructions for use manual and use accordingly. But anyway, this one is ACTA Avant provides high operational security. It prevents the column from coming together, prevents the pressures regulated better, and everything. You can use bigger columns. In this case, they are using high scale columns that can scale up. It can go up to 2 liters columns in terms of resins, and can handle high volume columns with maximum precision. So ultimately, what you guys want to do is you want to screen and identify a target molecule that you want to produce in more bulk volume, right? So ACTA Avant system is basically the pioneer in method optimization that can help you scale up to further ACTA and bigger resins, such as ACTA Pilot, ACTA Ready, and finally ACTA Process that can process up to 2,000 liters of resins and samples that you would after you optimize, finally scale up. Okay, the software you use is Unicorn, which is very functional and scalable, and your method can transfer from one Unicorn to the other one. Plus, the software that ACTA uses are all the same software. All you need to do is import and export. You can reproduce your data from one module to the other one. Okay, Unicorn is quite fast and efficient software, and yeah, so that can identify it has an inbuilt library of pre-packed resin that helps you check your resin inside the library, and that automatically pulls out the number for the pressure parameters like pre-column, post-column, delta column. All the pressure parameters are set there. If you choose pre-column, if you choose a pre-column conditions or pre-packed column, it will automatically bring the numbers because the database is uploaded in Unicorn and that you can choose and make. But of course, you need to read the instruction manual to clean up the column regularly and set a protocol for cleaning up and regenerating the column as well. Sure, you can always upgrade your AKTA to add additional functionalities. Even design of experiments is possible with your Pro, but you need to upgrade the system. You can always, you can upgrade your old Explorer to AKTA 1, AKTA Go to AKTA Pure L to M, and everything it can be done and customized. It allows you all to basically customize your AKTA according to your protein samples. We have something called AKTA Club. AKTA Club is it's a news feed where people, the pioneers of the AKTA, will be posting messages on the upgrades, on the updates, on the new software addition features that you can do, how to utilize the features in AKTA that is part of the AKTA Club. Please sign in as a member. You will basically get free content and online courses for improving your ability to one step more towards efficient purification, better expert, better resin selection, and also helps you choose the right consumables. That's what we want. Should not choose the wrong consumables, so we should be able to order, and then lead times is a problem. So please sign up for this club. You will receive updates, newsletters, and tips as well as purification. Okay, so that is positioning your AKTA based on your requirement. Any question for why we use AKTA Go, AKTA Pure, Micro? How do you use? How do you distinguish? Any question? If you don't ask question, I'm going to ask you question. Okay, no questions. So I'm going to ask questions. Yeah. That's it. If you're first, I will give you a sample volume. Okay, number. Tell me which AKTA you use, or sample volume configuration. Okay, I have a sample volume of 40 ml. Okay, I want to do a gradient elution, buffer A and B, ion exchange. My protein binds to the column. I dilute out in buffer B. Okay, and I know the elution condition at specific elution volume. It comes up. Okay, 40 ml volume, and the volume of my resin maybe 25 to 30 ml, no problem. What AKTA will I use? I know the elution condition. My sample volume is 40 ml, and I have 25 ml resin. AKTA Go was one answer. 40 ml. Very good, very good, very good, very good. Yeah. Okay. I don't have. I don't know. Use AKTA Go. Any more response? Any more AKTA that you think that? No, the question is what is the appropriate AKTA? Even AKTA, of course, AKTA Pure can do many things, but is AKTA Go can do something? Yes, correct. AKTA can do it. So you are the first one to get this right. AKTA can do because your sample volume 40 ml is high and optimal because you know the elution conditions that you can look in a specific concentration. Okay. Okay. Question 2. I have a sample volume of 20 ml. Okay, in a resin that is 25 ml, and I'm going to use a hydrophobic interaction. But what I don't know is what is the salt that is better for my elution and binding to the column and elution, right? So I'm very confused between ammonium sulfate and other set of salts, maybe sodium chloride also. I'm screening different salt conditions. I'm screening different salt conditions, sulfate, chloride, and chloride, and the air salts. And I don't know which is working, but I want to optimize this week. Okay. And I have a sample of 20 ml and I have 25 ml. What is the system that we use? Screening buffer conditions. Let me take other answers. Who can do? How many inlets your bow has? One buffer. Here. How many inlets that you have? How many inlets or buffers at your you don't know? Okay, so let's ask, okay, fine, on the specific numbers, fine. But you assume there's more than one inlet, right? So screening requires multiple inlets because you can screen buffers, because you cannot do one by one. If you can also do this in Application, but it will take you more than you need to do there multiple screening, right? So after pure is the answer. So you are like, so he's performing really well. Today, that's performing. And after pure can help you screen buffer conditions, screen pH conditions, screen column also. There's something called scouting of columns, you know that, right? Scouting of column is secret. One wall will have 10 columns. We have 5 column walls. So 5 column wall will scout for columns, called column screening. You can do that with pure. Okay. Go is not go. You can do this, but you have to do one by one. Buffer one, you see the not a problem, you can still do it. But active pure can give you a boost. Okay, so active pure is a great response. And finally, one question. I have 100 microliters. Okay, and I want to separate it by SAC. Right, I'm also think of it 100 microliter sample. I want to separate by SAC. Of course, there's other steps, exercising everything. But anyway, ultimately, I want to use 100 microliter sample in SAC. I want to get the protein as pure as possible. I have a super X column. I want to separate. Which ACTA will use? 100 microliters sample. Yeah, I will answer from micro. 100 microliters of sample. SAC, which ACTA can use. Okay. Other options? Okay. What do you think, micro? You need to say that after pure therapy, can do, right? After go can do. Can do, right? So after pure micros are working. Right? But why is after micro there? Because after pure and after go has the same so as far as you have the 100 microliter loop, you inject to products and then use after goes. So micro is not the answer, not the only answer. It is also micro. Right? So when is micro necessary? When is micro necessary? Sorry? What is it? It's true. Talking about technique 30. Right? Less than 100 microliters, like very less, like 30 microliters or 20 microliters, you have sample loop. So if you have 100 microliters, you can always inject the active pure the lowest and then flush the buffer. The problem will go and pure. Those sample volume is 100 microliters. is you will use sample because the cubates have a higher volume, high chance of cubates and retain sample. After pure micro, the cubates are so very small. How small is 0.1 millimeter diameter. So as a lowest you can go, which makes your tubing very small. Tubing very small, the sample take longer time to run. Longer time to run, better separation. Right? So after go and after pure, but its resolution cannot be. Right? So the efficiency is still lower in terms of purity. So that's why we need after. Okay, after pure micro smaller in inner diameter, and then smaller volumes, less loss. So the tubing ratio, sorry, loss is micro. And what is the problem with micro compared to pure? Yeah. Any challenge with after pure micro compared to after pure? Any challenge when you do this? Think of it. We go to hands-on later, but I'm just asking you. What is the note that you have to have in mind when you want to use after pure? Sorry? Fluoride because you are almost there, but you should. Fluoride is the effect. But what is the cost? What is the cost that makes you run a fluoride? Pressure. So whenever the cubates are very small, it will create high pressure because you are actually in the whole thing in small volume. So pressure is really high. So pressure goes up very fast. And then high chance that your alarm keep beeping all the time. So this you need to make sure that you run at a lower flow rate, which means lower flow rate it makes you stay in the lab for a long time. So that is a challenge. So make sure that you plan your experiment. So low flow rate make you guys to more time in cleaning, running samples, equilibration, and everything. Because better resolution will take. So that's a challenge. So we need to plan experiments with active micro, and also micro. There's high chance that your samples are reciprocated inside. Cleaning is a bit of a challenge for a long time. Right? So we need to prepare and expect high pressure for that. Running samples of lower pressure, lower flow rate. How low lowest we can do? What is the lowest flow rate we can? 0.45, 0.3. I've seen 0.45 as well. I suppose, but so in that case, 0.75, 0.5, 0.1, and the last one. So cubates are basically the diameter that your buffer gets. So dead volumes will be larger. So we can see that in the hands-on, so we can challenge it. Okay. Also the flow rate of the pump very kind of minutes for that. So that is very important. Make sure that you can handle the resin. If you want to pack big columns, if you want to pack the order big columns of the order, so you need to pack at a high flow rate more than 25 minutes. I think for your scale of application, 25 minutes for running your data process. So first choose the resin, first know your sample, choose the resin, just resin volume, resin and resin volume, and go for right ÄKTA. Okay. So based on panel application, you choose the right ÄKTA. But if you're taking step as 3 steps or 2 steps, you can use the first step in ÄKTA go or your resin. But the final step has to be micro, you need to minimize the sample complexity. That's the final step, or one step to use. Okay, rest of the routine repeats or optimization So, next, that's the positioning system. Any questions on this? Please.
Actually, for several cases, we have to do purification and purification for two different protein. So both are RNA, and the volume may be 100 or 150 ml. And so sometimes we do one protein purification RNA in Tanggal, and another one is in Apil with column. So essentially,
Like you have a factory.
yeah, it's like so my question is, since we don't have the super loop, we can we just use the inlet for the inlet of the appeal for the protein? Yeah.
But you don't have a
Oh, that's some.
I think using inlets for sample is not possible because inlets is for longer. Sample is for basically having the injection wall that goes to the fund. So inlets, basically for using inlets, for samples will need something else. Because the problem will be very long. So it's not very nice. So what is the following? What is the following in that?
Let's say
Wow, what is the sample of this?
is it can be damaging the sample pump or just sample loss?
I was recommending getting a sample pump.
The sample pump is it will be damaged if we use the buffer pump.
Yeah, it's not damaged. Oh, nothing has been damaged, but you do something. You do something. It's very hard to cover sample because of the but the buffer is basically the pump, right? The pump is huge. So if you want to push the sample inside, you use sample. Why do you want to do this?
So it's okay if you use sample, but again, my question is, I'm not going to damage the pump. Is it so?
No, sample won't be damaged. What is your sample? Oh, the true sample. No, you can't use license. You need to clarify that. You can't use cell license. Cell license are having a lot of various. So that's what I do. The pump can block.
So even I use some saline other.
No, since we preparation, okay, can use it in but generally, exactly, no one not ready to apply sample in a public. So even if you want to use it for applications, only about yes, we can get super or do a multiple injection. Like we have to try them. So let's think of the cycle sample sample. So inject another multiple times. You see another you know, that something for program. Who has it? You said. You said, go, I need to go, kind of the sample. You said, go, you have another thing. In one shot, you can do what is the volume of this? Listen, 5 minutes. Every in one part, how much sample you can? Okay, 5 ml. Okay. I think you can take up to 10 pounds. 50 ml. You can use it half a row. Inject 50 ml in one shot. And if 50 ml is very high, you can do 25 ml 4 times. After go. Afterglow cannot do HIC. That's for sure. Afterglow cannot because ammonia solvent and afterglow doesn't. I see. It's not only I don't know it like but you can. So in your condition, I will use afterglow. You do that. Wonderful. Okay. That's one. Yeah.
That's all. So when we have sometimes we have evolution step, if the evolution protein is very low, the absorban to 20 also detect the idazole.
Good question. Good question. Okay.
You can different question.
What is your?
So essentially, if the target protein is so low, so the peak is not edible because of the absorbance of the idazole. So isn't anything to solve that? I mean, if the is very low, protein cannot it cannot be seen.
Yeah. So what is the okay? It's a good question. It's a very good question. Imidazole and chronic brain has have a constant increase to it. Imidazole, for example, I saw UV doing me from 0 to 100%. Also do imidazole from 0 to 100%. So the way to do this is you can't see the peak, but you are purifying. So you need to put in estimation. So yeah, your chromatograph will have that cannot be solved because we have only detectors of purity. One thing to do is Periset tag. The current tag only you use another detector. If you want to see the protein, put a tag on it, CFP tag or red tag. Use a different wavelength, you will see a protein. So if you want to create a chromatograph, see only a protein. I will use the protein with the fluorescent tag. Yeah. But if your purpose is just to purify, then you see. Is you do the gradient and then you do sample estimation. You need to calculate the power by yourself, either by Liza or because UV and imidazole impact. One more thing is that if you still want to see the protein, you can resort the imidazole from all the parts of the again. So you will see only remove the imidazole. Then what the peak you see on the only protein. Yeah,
I will use the so I mean, which one is more efficient by using the ultrasonic group or by using the dissolving column transcript?
No, this is ultrasonic. Oh, Microsoft. The other option is what is this one is okay. Ultimate support for the so these two products are not the same. So desorting is to remove salt. No, can invisible salt, right? So desorting for a purpose is to remove salt. But ultrasensitive is microset or microset.
Yeah.
The other one is to remove proteins above a specific amount. Two different products. They can't compare. So you see what I mean? Okay, if you want to use middle class, you can use desorting for the problem. But if you want to use if you want to remove middle and sorry, if you want to and documenting both can do something. But this one can have some molecule of things after certain time. So these two products just remove the salt, that's what you what can do. But what do you want to push to centrifuge? Then we have something following. It's only salt removal. It's both protein and salt removal I use that. Yeah, one. So two different applications. It's very subjective. It's very subjective. One company is not have the other company. More questions from you? In that case, we will wrap the session, then we will meet. What is the next slot? Yeah, thank you. So I will stop sharing my screen. Oh, I have more questions.
Momen tipe akta sistem internal.
Oh,
Oh iya, Mbak. Ini pertanyaannya lebih ke Elsa soalnya. Nggak ngerti.
Apa aja di Genomik?
Di Genomik apa di PPU? Pertanyaannya tipe akta yang berada di lab Genomik sama kolom apa saja yang tersedia dan bisa diakses menggunakan Elsa.
Oke. Oke, jawab mana? It's like for internal, like each type of system that we have. Like what kind of column that we have.
Oke, untuk materi dari Satya. Oke, jadi itu materi dari Satya. Yang pertama adalah itu tahapan preparasi yang salah satu yang krusial sebelum kita masuk ke arah protein modifikasi. Lalu yang kedua itu memang lebih intens pada apa yang langsung perihal dengan kromatografi sistem yang akan kita hands on. Jadi secara keseluruhan sudah sebagian setengah jalan dari proses modifikasi sudah kita pelajari secara teori. Selanjutnya adalah setelah makan siang kita langsung hands on mulai dari tahap preparasi yang tadi. Jadi tahap penerapan dari materi tersebut. Jadi harapannya ketika nanti hands on ada pertanyaan atau ada challenge yang memang sudah kebayang, kita sudah pernah dilakukan, dengan senang hati silahkan bertanya. Bapak dan Ibu jangan malu-malu. Jadi harapannya kita lakukan hari ini sampai 3 hari ke depan itu benar-benar bisa membantu proses riset dari Bapak dan Ibu sampai memperoleh hasil yang sesuai dengan harapan gitu. Kalaupun ada challenge 1, 2, 3 itu pasti challenge-nya akan beragam untuk masing-masing Bapak dan Ibu. Jadi jangan segan untuk ditanyakan. Kurang lebih seperti itu. Kalau ada pertanyaan bisa reach out nanti ke saya, ke Satya, ke tim ataupun ke tim Mbak Seni, Mikrobiologi dan rekan-rekan yang lain karena ya kita cukup intens untuk komunikasi. Jadi Silahkan saja nanti ditanyakan. Terima
Terima
kasih
kasih
semua.
semua. Thank you.
Saya kembalikan ke Mbak.
Thank you, Ms. Vidya, for leading the session, and thank you, Mr. Satya, for the interesting presentations. Ladies and gentlemen, now it's time for lunch break, and we will continue the agenda at 1 p.m. with the presentation from Cytiva and Laborindo Sarana. Thank you for joining this webinar, and see you in the next event. Wassalamualaikum warahmatullahi wabarakatuh.
Thank you.